Hello, my name is Timothy Britt, and I will be giving a synopsis of the podcast Bacteriophage Blocks Bacterial Bouncers for Dr. Santos's microbiology class at UNCP. This podcast was originally made by Jesse Noor, where he explained how a new species of bacteriophage was able to defeat science's most advanced defense systems. I hope you enjoy it. To begin, most people are probably not sure what a bacteriophage is. The definition of a bacteriophage is a virus that infects a bacterium by reproducing inside of it. The relationship between the bacterium and its bacteriophage can be described as a parasitic relationship. As you probably know, a parasitic relationship is a relationship in which one organism infects a host organism while the parasite benefits from the host is being harmed. Moreover, scientists have developed defense mechanisms such as the CRISPR-Cas9 system to help defeat these parasites and viruses. If the virus can overcome the defenses such as the CRISPR-Cas9 system, which we will talk about more in the next slide, it can spread rapidly, taking over the host. However, there are some limitations to the bacteriophage. It often takes the sacrifice of multiple phages before one is able to successfully infect the host. Like I said before, the CRISPR-Cas9 system is a type of defense that detects and chops up the viral genomes. Although the CRISPR system is supposed to be able to adapt to new phage mutations, there are still some viruses that are able to defeat the system. Now we're going to take a look at the video that further explains how the CRISPR-Cas9 system works. Now you can see that the CRISPR-Cas9 system works much like a cut and paste tool. Broken down, the system has two components, a Cas9 protein that works to cut the sequence, and a guide RNA. The guide RNA is attached to the Cas9 protein and is able to introduce it to the target sequence. Scientists are able to use this system to modify genomes by altering or adding new sequences. Like I said before, the CRISPR-Cas9 system is a type of defense that detects and chops up the viral genomes. Although the CRISPR system is supposed to be able to adapt to new phage mutations, there are still some viruses that are able to defeat the system. Now we're going to take a look at the video that further explains how the CRISPR-Cas9 system works.
In a document, if we suspect we've misspelled a word, we can use the find function to highlight the error and correct it or delete it. Within our DNA, that function is taken on by a system called CRISPR-Cas9. CRISPR is short for clustered regularly interspaced short palindromic repeats. CRISPR consists of two components, the Cas9 protein that can cut DNA and a guide RNA that can recognize the sequence of DNA to be edited. To use CRISPR-Cas9, scientists first identify the sequence of the human genome that's causing.
Now you can see that the CRISPR-Cas9 system works much like a cut and paste tool. Broken down, the system has two components, a Cas9 protein that works to cut the sequence and a guide RNA. The guide RNA is attached to the Cas9 protein and is able to introduce it to the target sequence. Scientists are able to use this system to modify genomes by altering or adding new sequences.
A health problem. Then they create.
Going off of the previous slide, I mentioned that some viruses are still able to defeat the CRISPR system. Scientists have the ability to engineer many different types of CRISPR defense system. So this gives them a better chance of finding a method by which to defeat the pathogen. However, in one particular study, scientists found that one particular phage was so strong it was able to survive even when combated with all known CRISPR systems. One of the better known viruses for doing this is a phage that commonly infects the bacteria Pseudomonas aeruginosa. While this bacteria is to have many different phages, one of its strongest is known as 5KZ. 5KZ is a versatile microbe that causes serious infections, and when scientists engineered multiple types of CRISPR defense systems to fight off the phage, 5KZ proved to be stronger and still survived. Experiments were conducted to see if the CRISPR system could fight off a 5KC phage that had infected the host. However, the 5KC still survived even against multiple CRISPR systems. With the 5KC phage possessing broad resistance to all CRISPR systems, scientists were faced with looking to other options.
A specific guide RNA to recognize.
While CRISPR defense systems and genome targeting are ways phages can be fought off, genome editing can also be accomplished through restriction systems. Restriction enzymes are able to alter nucleic acid sequences by going in and cutting specific restriction sites. At the restriction sites, enzymes cut the DNA into two places, the sugar phosphate backbone and the DNA double helix. Today, restriction enzymes are commonly used to cut out DNA sequences and make genetic engineering possible. But aside from this, restriction enzymes are very useful in many other ways. One of these ways being protection against infections and viruses. When a cell becomes infected, restriction enzymes are able to locate the viral DNA sequence and cut them out so replication is put to an end. For example, specific sequences made up of nucleic acids such as GTTC could be targeted and cut out. With the viral genome cut out and no longer able to replicate, no further damage can be done to the host.
That particular stretch of A's, T's.
While studying the well-known microbe Pseudomonas aeruginosa, scientists found that one of its phages, 5KZ, was able to infect even with many different types of CRISPR systems in place. This particular microbe can be especially dangerous, so scientists conducted experiments to see just how the phages were thriving and figure out a way to stop them. After successful infections of the 5KC phage, even in the presence of defense systems, scientists knew something was special about its ability to survive. The phage was able to inhabit and replicate within a host not only in the presence of the CRISPR defense system, but restriction enzymes as well. After inspection, no anti-CRISPR genes were found in the phage, so its structure was analyzed. It was discovered that it had the ability to survive because of a specialized, self-constructed protein structure. Inside this nucleus-like protein shell, which is made up of a cytosilicon components, DNA replication is able to take place in this sort of safety chamber. With such a structure giving the phage free range to self-replicate, the phage has no problem surviving, even in the face of advanced defense systems. However, when the DNA was extracted from the protein shell and studied in vitro, it was degraded in the presence of CRISPR. This told scientists that the phage itself was not immune, instead, it simply had an advanced defense system.
G's and C's in the DNA.
After the discovery of the protein barrier, scientists had a few key questions to consider before conducting more experiments. One of those questions was why and how phage DNA was not protected in the absence of a protein barrier. And if DNA was vulnerable outside the shell, how were scientists supposed to get inside or deconstruct the safety chamber?
The guide RNA is attached to the.
Without knowledge of how to deconstruct the safety compartment, scientists experimented with ways to bypass it. Initially, they tried attaching a CRISPR-Cas system to one of the proteins that was included in the construction of the chamber. However, no matter where on the protein the defense system was attached, it was too large to pass through and did not affect the phage. Next, they decided to attach a smaller protein in hopes that it would be small enough to pass through when attached to the pre-existing protein. This smaller protein, the restriction enzyme EcoR1, will be used to enter the safety chamber to cut the nucleic acid sequence. Luckily, the bound enzyme was able to enter the safety chamber and cleave the DNA sequence. This process was successful and reduced phage replication by nearly 500 times. In addition to the EcoR1 enzyme, a fluorescent protein was also attached to track what was taking place within the structure. And somehow, the presence of the fluorescent protein allowed for easier disruption of the shell construction process. While this fluorescent protein was initially only added to tag the EcoR1 protein, it actually ended up adding to the degradation of the phage.
The DNA cutting enzyme Cas.
Normally, the CRISPR defense system used in the Pseudomonas species cuts DNA to target the genome. However, a new type of CRISPR engineered technology has emerged that allows the system to utilize RNA. By targeting RNA, the need to enter the shell is eliminated. As the messenger RNAs exit the structure to be translated into viral proteins, they are compromised. With altered mRNA no longer able to translate the viral genome, CRISPR RNA is able to stop the replication process before it even begins. This means that the virus is no longer able to replicate and is thus unable to further infect the host. While CRISPR DNA targeting focuses on controlling viral infections, CRISPR RNA targeting is able to focus on eliminating expression of viral genes firsthand. In the diagram, you can see that the 5kZ nucleus-like structure excludes multiple case proteins and different types of restriction enzymes. However, case 13, the Listeria engineered CRISPR, is able to target mRNA, which is in the red. During exportation from the structure, you can also see that, as previously mentioned, the binding of the EcoR1 enzyme to a pre-existing protein is successful.
9. And then this complex is introduced.
As experiments have shown that phage DNA is more vulnerable when exposed outside of the protein shell, theories as to why this happens have emerged. For example, scientists wonder why the phage is able to survive before it has constructed its shell. Some theorists suggest that other protein protectors are present to defend the phage while construction takes place. Some theories also suggest that phages are able to take advantage of the host for protection and benefits. Again, these are only theories, and the Pseudomonas species and 5kZ phage are still being researched.
To the target cells.
With the discovery of how to defeat the 5kZ phage, opportunities for many other advantages arise. Not only does this new information present potential answers to some of science's questions about nucleus origins, but it also can be adapted to make new strides in the medical community. For example, doctors can now target other pathogens that have strong immunity. They can also learn to protect useful microbes from phages with unique defense systems like 5kZ. In our body, we have multiple helpful microbes. When bacteriophages infect these microbes, our body loses all of the microbes' benefits. This is why it is helpful that scientists have discovered new ways to fight off these harmful bacteriophages. The new discovery also raises a few important questions. What does it tell us about the origin of eukaryotic nuclei? This is the possibility that during the development of the nucleus, the cell adapted a similar type of safety chamber to protect its DNA.
Locates the target letter sequence.
With the discovery of how to defeat the 5kC phage, opportunities for many other advantages arise. Not only does the new information present potentially answers to some of the science's questions about nucleus origins, but it also can be adapted to make new strides in the medical community. For example, doctors can now target other pathogens that have strong immunity. They can also learn to protect useful microbes from phages with unique defense systems like 5kZ. In our body, we have multiple helpful microbes. When bacteriophages infect these microbes, our body loses all of those microbes' benefits. This is why it is helpful that scientists have discovered new ways to fight off these harmful bacteriophages. The new discovery also raises a few important questions. What does it tell us about the origin of eukaryotic nuclei? There is the possibility that during the development of the nucleus, the cell adapted a similar type of safety chamber to protect its own DNA. Well, this is all the time we have for today. I hope you have gained further insight as to what bacteriophages are and what some are capable of. If you would like to learn more about the topic, you can visit the link to the original podcast in the description provided below. I will also include the link to the original findings in the journal that documents the initial experiments. Thank you for watching.
And cuts the DNA. At that point, scientists can then edit the existing genome by either modifying, deleting, or inserting new sequences. It effectively makes CRISPR-Cas9 a cut and paste tool for DNA editing. In the future, scientists hope to use CRISPR-Cas9 to develop critical advances in patient care or even cure lifelong inherited diseases.
Now you can see that the CRISPR-Cas9 system works much like a cut and paste tool. Broken down, this system has two components: a Cas9 protein that works to cut the sequence, and a guide RNA. The guide RNA is attached to the Cas9 protein and is able to introduce it to the target sequence. Scientists are able to use this system to modify genomes by altering or adding new sequences.